Good evening to all of you. We are back again with another exciting session. And I, Ajay Jha, on behalf of ACS India team, welcome you to today's ACS Science Talks session. Today, we are pleased to have Professor Sudipta Maity with us. He's a professor at Department of Chemical Sciences, Tata Institute of Fundamental Research, Bombay. He received his PhD in biophysics from University of Pennsylvania, USA. Thereafter, he was a postdoctoral fellow with Professor Matt W. Webb at Cornell University. His research group is actively engaged in the study of protein aggregation, protein misfolding, and neuronal interactions, a glimpse of which he'll be sharing with us today. Professor Maity has numerous awards and accolades to his credit, to mention a few. Wellcome Trust Senior Overseas Research Fellowship in Biomedical Sciences, Award for Excellence in Teaching by University of Rochester, SICO Award from the National Academy of Sciences, India. He's the co-founder of the Fluorescence Society of India, as many of you know. And he's the senior editor of JPCB, EAB member of ACS Chemical Neuroscience, and also serves as the editorial board member of several scientific journals of international repute. So before Professor Maity starts, I have few instructions for the audiences. We request the audiences to post their questions into the Q&A tab or to the Zoom window. And kindly do not put questions into the chat box. In case of any internet issue, please stay with us. Now, I would like to invite Professor Maity to deliver his talk. Professor Maity, please.
Thank you very much, Ajay, and a very good afternoon to all of you. I know it's a holiday, and you guys have turned on your monitors just to listen to me. I do appreciate that. Let's chat some science. I like to take it slow and keep it at a level that everybody should be able to understand. But at the end, we'll have the Q&A session. So if there is something that is not understandable, let me know. So yes, so thanks to ACS, especially because my association with ACS, as maybe for most of you, is long as author and contributor. But as you start getting gray hair, you get associated in different capacities. So I've been for a few years the member of the editorial board of ACS Chemical Neuroscience. However, just yesterday I took over. I joined as a senior editor of JPCB. So glad to have this association with one of the premier societies of chemical societies of the world. And I love ACS journals because, first of all, it comes from a society and not a private company. And there is no publication charges. So you should know about all those. And as a senior editor of JPCB, if you have papers that our JPCB has the right audience, mainly biophysics, biophysical spectroscopy, etc., do send it to us. Some of those will land up on my desk, and I cannot ensure that they will get published, but I can definitely ensure you'll get a fair and rapid review. Okay, so the date is special, of course. So let me actually pay homage to the person, the reason for which today is a holiday. In between running and organizing a revolution, running a big political party, responding to famine, responding to violence, responding to calls for help, philosophical discussions with Tolstoy and Tagore and others, this man did find time for science or to meet his scientific curiosities. Is one of these rare pictures that I really treasure, because I also do a lot of microscopy of Gandhiji peering through a microscope. Apparently, this is something about tuberculosis that was on the slide, and which is, of course, was very strongly in his mind. So fundamental science is something that is inseparable, whether it's from any country's psyche, whether it is a developed country or it's a developing one. Tuberculosis, of course, has been a scourge for a long time for us. And the newest one, COVID, the reason that many of us are partly at home, is something that will also hopefully be solved by science pretty soon. I mean, there are in fact dates of months or maybe a year by the time we get a vaccine. So hats off to science and scientific research for that. So I'll tell you about Playdough proteins, okay? But before I get to that, I'll tell you about the questions that we are passionate about in our lab. And I say we, it's me and a few of my students, and there is no, it's all a horizontal group, and we really, they mostly break their backs. I also try to burn the midnight oil, try to get to the basics of fundamental questions. Some of the things relate to protein aggregation and toxicity that's related to it, and I'll talk about that today. But some other things are actually not related to it. I won't get to talk about them. So let me tell you a little bit about them. We are interested in neuronal signaling. And in neuronal signaling, how especially monoamine signal between neurons, help signal between neurons. We're interested in developing a lot of photonic tools, which is especially applicable to biology, including single molecule tools, which have been commercialized. And lately, we have been very interested in neurotransmitters again, but not their standard mode of signaling, which is through a neurotransmitter receptor, but how these small molecules can actually possibly signal even without a receptor by changing the properties of the lipid membrane. So these are things we would not be able to talk about, but you're welcome to read some of those things if you're interested, or ask me questions later. So come back to Playdough proteins. Everybody has seen a Playdough, right? Here's a Playdough. Okay. The good thing about a Playdough is it can give it any shape. It's nice and round. You can make it flat. Maybe if you like a hole in it, you can put that hole in it. You can make a heart out of it, whatever you want. A same piece of matter, you can give different shapes. The question I'll be asking in this talk is, can you do that with proteins? And if this was a live audience, I would say, raise your hand if you can do it. I can see you very well. So I will give the answer myself, but after this little movie. So look at a protein folding. Proteins are just a heteropolymer, okay, completely inanimate, 20 different blocks that can hang together, and 20 different types of blocks, of course, many of them, and see what happens if we drop them in water. Okay, there is a slide, a movie borrowed from UIUC. Look, so this is a protein, and that's the extended part. You can see the string as the extended part, and it just dropped it into water. And this is a computer simulation of what's happening. You see what's happening. That protein, completely inanimate, is taking a definite shape. The shape that is predetermined, because that shape dictates how the protein will work. It's still trying to get there. It can't. It doesn't know where it has to go, but finally it does. Finally, it does get there because of thermodynamics. This, to me, is the wonder of life. Inanimate life becoming real living object, because this is really the basis how your DNA code gets translated into something which is this heteropolymer, which becomes a functional machine. There's a one-to-one correspondence. Okay, that's the standard model of biology, sometimes it's called, that you have DNA, you have transcription, which makes RNA, messenger RNA, then you have the sequence, which the ribosome makes, and it folds by itself. There's a single best example I know when somebody asks me, do we need a supernatural being to explain life? This is why we don't need it. This is how the basic thing of life forms. The functional machines, and it's completely encoded in it. Of course, we have been trying to understand it. Much of it is understood, some of it is not. But it also tells me that if you have a sequence of a protein, you have a shape. Because that's how life progresses. Life goes between generations. You have the DNA, the sequence of the protein, which is this, and then the three-dimensional architecture is encoded in that. This is the problem of protein folding. And this is why a protein is not a Play-Doh. You cannot give it a shape that you just want, or you cannot just put it somewhere and hey, hope this time it will take a different shape, and pick it up, unfold it, put it again and say, oh, this time maybe it will take a different shape. No, the thermodynamics and the sequence ensures that it will get to a proper shape. Otherwise, life would not exist as we know it. Okay, so you can't make Play-Dohs out of proteins as we understand protein folding for standard proteins which fold well. Okay, so how does the standard protein fold well? Well, this is a good example. There are various forces at work, but one of the major forces at work or interaction at work is something called hydrophobicity. Okay, its origin is in thermodynamics, basically in the entropy part of it. And what happens is that proteins, to a large extent, can be understood as a two-color code: hydrophobic residues, hydrophilic residues. Okay, and here's blue hydrophilic residues, yellow hydrophobic residues. Some are in between; those are white. This is myoglobin, one of the proteins, common proteins in a muscle. And it shows that when it's dropped into water, it gets this shape because it tries to wrap itself up into hydrophilic residues and bury its hydrophobic residues in the center. Here is a cut through the center plane, and you see most of it is yellow, but that's hidden from the solution around it, from the water around it. It's the surface which is blue, which is hydrophobic. So proteins have been organized so that they can self-assemble to a particular structure, minimizing their energy, minimizing the exposure of hydrophobic residues, and maximizing the exposure of charge and polar residues to water. And that gives finally one unique shape where the energy is a minimum. Okay, and this is where the one-to-one correspondence comes from. Oh, well, I mean, you think you understand nature, and nature throws a curveball at you, and you certainly understand, well, that's not how it should work. So here is a protein, okay, or a class of proteins, which actually does not work like what I just told you about. They are proteins which can take different shapes, okay? How do they do that? Is everything that I have just said 2 minutes ago wrong? No. Think about it this way. If you snip a small peptide, small parts of proteins are called peptides, or small length proteins are called peptides. So snip it from a protein, would it be able to fold? The answer is most likely not, because you need to have enough of the hydrophilic residues to wrap yourself up and to hide your inner core, which is the hydrophobic residues, in water. If you don't have so many parts, then you will be frustrated and you will not be able to wrap yourself up and be in water. So here is an amyloid protein. And so if you have a small part or a small peptide, here is amyloid beta, which is a cartoon representation of amyloid beta, which is a protein that is processed by, it is there in the body, a peptide, which comes out because enzymes cut a particular protein, amyloid precursor peptide, a small part of it, about 40 residues long part of it, 40 blocks long part of it, and it cannot fold by itself. So as it is in water, it has some random shape. But there is a way out for it to take a particular structure. The way out is it can gather together. There are hydrophobic parts of it, can gather together, and what one particular monomer cannot do, 10 of them may be able to do. And this is what is already something that nature does. Unfortunately, to devastating consequences. When amyloid beta aggregates, you get Alzheimer's disease, something that whose cure we do not know. It's a very common disease of the old age. It's not the only one. There are several such diseases whose origin is in the misfolding and aggregation. And there's a huge race on to understand what are the structures, these things which are roughly unstructured, what are they making when they're coming together. So this is, if you look at amyloid beta aggregates under a transmission electron microscope, you'll see these fibril-like things, fibers. Okay, so they do make some particular structure. They are not like this. In fact, that structure, the atomic level, gets refined and refined and refined. The newest one is not yet properly published. One of the pioneers of the field, Rob Tico, and his co-workers have put something in bioRxiv just a few weeks ago. This is somehow lots of these stack together and make this fibril, and this is their latest idea about how this happens in somebody's brain, which the patient who has Alzheimer's. Question is, does that explain how they are toxic? Okay, you have a fibril, but I just said that this makes the protein toxic. Is this structure something that tells me how it will be toxic? The answer is no. Because, and this is a big if here, but the best guess we have is that before this guy, the monomer becomes this large mer, it forms an intermediate level structure. Here it's a Playdough nature with a hole in it, because this is a membrane. Think about the layer of lipid bilayer that covers the cell, and it punches a hole through the membrane that is permeable to ions. That's bad news if you're a neuron, because you can communicate and live only because you can separate out ion gradients on two sides of the membrane. One is the cellular side, cytoplasmic side, other is the extracellular side. And these things punch holes in the membrane. That's the best guess, and make something like this. This is a cartoon drawn inspired by another protein whose structure is well known. And the guess is that amyloid beta makes something like this. So this is a Playdough protein, monomers. This is some sort of beta sheet structure. This is called an antiparallel beta sheet structure. If you look at the secondary structure, parallel beta sheet structure here. This is a beta barrel protein, sometimes called a porine family of proteins. That's our guess. And this is an antiparallel beta sheet structure. So this structure doesn't explain how this can form. And over the last 5 or 6 years, we have been trying to look at whether this is just our imagination or this can exist. Okay. And I'll take you step by step through, just in little bits. Every step came with a lot of effort by most of the time by inventing a new method of spectroscopy or microscopy. But I think we have the answer to a large extent now. So these are the two things I'll tell you about. First is when nature plays Play-Doh with proteins, which is these amyloids. The few questions I'll ask is that for amyloid beta, is the oligomer different at the secondary structure level? Because it has to be antiparallel. Can we experimentally see that the oligomer is different? Does it maintain that same structure in the membrane? It forms in the solution. Can it maintain that in the membrane? Does it have a defined non-random orientation in the membrane? That's very important if you're a channel. If you go every which way, you're not a very good channel. You have to go right through maintaining your orientation. Does it have a defined number of strands? If it has to be a proper structure which passes, some people think it's a calcium ion selective ion channel. So you have to form an ion channel from different parts. You have to have a certain size. So for that, you need to add up, aggregate together, maybe 3, maybe 4, maybe 5, but not 300. So do you have a particular number of strands, number of monomers in that oligomer which sits in the membrane, if it does? And then, as we will see, there are still unstructured parts. Not every part of the protein becomes structured. We ask whether the unstructured parts are good for anything. Okay, so I will quickly answer these one by one. And if I get time at the end, we'll ask a very different question, which is when we, scientists, play with Play-Doh, play with proteins as Play-Doh. Okay, and I'll tell you something about decoy peptides if I get time. Okay, let's start then. The first one is, can the secondary structure be antiparallel? Okay. As we said, the large fibrils, which you saw in a TEM, those structures are coming out, but for a long time it has been known they're parallel beta sheet, which is a kind of secondary structure, tells how different parts of the protein are hydrogen bonded together to give the overall shape. However, the porine structure, the pore kind of structure, is known to be antiparallel. So can this be antiparallel? This is very, very difficult to find out because the intermediate structure exists for brief time only at a very low concentration. So you need to develop techniques, which you did, combining fluorescence, single molecule fluorescence, and NMR to finally get to some idea of the secondary structure. It was enough. And this is where we were 3 years ago, 2017, and also JPCB, where we could show that, yes, it has an antiparallel structure. The antiparallel becomes parallel later, but the oligomers do have an antiparallel structure. Okay, so now the second question is, what is its conformation in the membrane? That one I just showed, which you did, started 6 years ago, and final answer came 3 years ago. Whether we can expect that to be the similar structure in the membrane. Okay, antiparallel, yes, that agrees with our idea of a beta barrel, but can it be sitting in the membrane? unlikely, you would say, because if you know a little bit about how proteins work, a protein structure which is something in a solution, it won't like to sit in the membrane, but if it does, you will see hydrophobic environment all around it. So most likely the structure will change. So we asked that question, and it was also an even harder question to ask. We had to invent a technique which takes nanoparticles and plasmonics. I won't be able to get into that too much, but these are effects of light fields, and how it resonates with the nanoparticles, which enhances or quenches sometimes the spectral signatures of light. So it enhances, for example, Raman spectra. That's what we used here. And we wrapped a membrane around a gold nanoparticle. The membrane is seen as little pale shadow, and this is the gold nanoparticles or silver nanoparticle, both will work. And we built an instrument which can look at these at a very low power, and we did surface enhanced Raman spectroscopy. Okay? And we found out that yes, these are still antiparallel as they sit in the membrane. It's our first crack at the problem, because till then we only knew, okay, it could be antiparallel in the solution. And therefore, it can look like, at least all we could tell is that of individual strands. Individual strands can agree with what a porine individual strand looks like. We still have to make sure that they go together in a proper orientation, and we have to make sure, are they just a jumble of things, 100, 200 such things, or just 3-4 proper structure? Okay, so this was good. This is where we were about 5 years ago. Then we asked the second question: is the oligomer oriented in some fashion in the membrane? Because if it's just a random jumble, it will go into the membrane any which way. We can hope for a ion channel kind of structure only when it has a certain regular orientation in the membrane. C terminals go in a particular direction, N termini go in a particular direction, etc. How do we tell? Well, plasmonics came to our rescue, and we developed another method, which is plasmonic quenching. So we wrapped again this lipid bilayer, as you can see here, around a nanoparticle, but this time we are doing fluorescence spectroscopy, not Raman. We put fluorescence tags one by one on the C terminus of the protein or on the N terminus of the protein. Now, as I said, Raman gets enhanced by the plasmonic field, but close together, if you're within a few nanometers, actually fluorescence gets quenched. That is, you get much less fluorescence. So that can be seen in something called fluorescence lifetime. So you can measure the fluorescence lifetime of these objects when, let's say, the C terminal is there only labeled, fluorescently labeled, or when the N terminal is labeled, and measure the lifetime and say, okay, what we found was that the C terminal is close to the inside the nanoparticle, which the lipid bilayer is wrapped around, and the N terminal is reasonably far from it. We couldn't put in numbers, but C terminal looked very close, N terminal is relatively farther out from it. Okay, if we put everything together, then with this technique, we finally said, well, it probably makes a structure which looks like a Z, okay, or an S, whatever. There's a C terminal inside, and there is an N terminal outside or hanging in somewhere, making a pore or something, but further away from the inner side of it. So this is at least satisfies our criterion that if it has to be a pore, it has to have a certain regular orientation. And we could show that, yes, it does have a regular orientation. So then what? The next question is it formed of a regular number of particles, number of monomers, or is it just a jumble? Okay, it's oriented. Okay, it's an antiparallel beta sheet, but it's like 50 things just coming together randomly. So we had to count how many proteins, how many monomers are there in one of these oligomers. How do I count? Because these are so small, you cannot see them separately in any kind of microscope on a lipid bilayer. Okay, so what we did, we went for single molecule counting. We wanted to count these things one by one, and there is a technique that can do it. It's called single molecule photobleaching. So you label each of your monomers with one little fluorophore, and then suppose there are 4 of these. So this cartoon shows imagine there are 4 of these, they're all bright and green fluorescing, then what happens is that in water, as you keep exciting them, they will bleach. That is, they will go off like a bulb, fusing. So 1 will go off, 2 will go off, 3 and 4, all will go off. You will see the brightness of individual clusters decrease if you are that sensitive. And one of my gifted grad students, Shrimli Dey, who has just graduated, built this instrument in the lab. So a lot of these instruments we built in the lab and showed that, yes, she could count. So here are some examples. Of course, she found a distribution of things. There are some monomers which bleaches in 1 step, and some other things which bleach in 3 steps, 4 steps, etc. You could do a statistics. And when you do a statistics, we found that, well, there are quite a few monomers, some dimers, some little bit of trimers and tetramers. But we also recognize this whole method, which was developed by others, has a flaw in it. So she recognized that. And luckily, we could come up with another method to correct for that flaw. I would not talk about it. But after correction, this is what the distribution looks like. Mostly, they are tetrameric. I've shadowed out some of the higher marks because the statistics is very poor. But if I talk about 1, 2, 3, 4, and many people think that it's only a few marks, not very large mark, there are other evidences for it, then it's mostly tetrameric, which actually will fit beautifully. Each monomer is a folded structure, so it gives 2 strands, and 4 of these will be 8 strands. An 8-stranded beta barrel is quite a common object, and this is what we find. Of course, the proteins that you have looked through, I just talked about amyloid beta. This was actually done in a sister protein, which is amylin, or something that causes type 2 diabetes, but the principles remain the same. Okay, so this is where we came. And the last question of the first part is, okay, so we have looked at order, we have looked at how they're oriented and organized, and you looked at how many of them are there. Is everything structured? And we did solid-state NMR, we already knew that things are not fully structured. A protein, many proteins, sometimes will have a little part of it which is loose, in the sense is not structured. And these proteins start with being completely non-structured. So it's not surprising that even after they aggregate together and find a way to properly fold, they'll still be unstructured parts. And something that, so we could measure that the unstructured parts is then the N-terminus, okay, near the N-terminus. Something told us that's really an important area, because if you look at human mutations, people who get Alzheimer's disease very early, there are people who get Alzheimer's at 40, 45, which is a very unusually young age. Usually you'll find out they have some mutations which make them more vulnerable to Alzheimer's disease. And some of these mutations cluster in the N-terminus. So it's obvious that though it is not structured, it probably has a lot of importance in how the toxicity happens. Perhaps it is free to interact with something else in the cell, which will make it toxic. Can we tell if it is interacting with something in the cell? I mean, the cell has 30,000 different things. It's difficult to figure out if in a real biological context, particular protein that you introduce in the cell will actually interact with something else. So we do a lot of microscopy also. So here we see something that is done by confocal microscopy of individual neurons. So one of these things is an individual neuron, and we dealt with 2 different proteins. One is the rhodamine level, R stands for rhodamine, amyloid beta 1 to 40. The one where we have cut off the first 9 residues. Okay, and we saw, yeah, it doesn't matter. So if you look at these, this is some other marker which marks the endosomal parts where this A beta goes to, and we put the peptides, whether they had the N-terminal or did not have the N-terminal, they all went in. It's not dark, it's pretty bright and red. And if we co-localize, we see that they're co-localized with transferrin. That means they're going to the endosomes. Fair enough. So that means at least it's not true that the cells do not interact with things which do not have the N-terminal. However, if you measure the toxicity, and you have separately measured that, it is known that the N-terminal, if you chop off, it's not toxic at all. So we did something called fluorescence lifetime imaging microscopy, again, partly home-built system, where you could see that if you put the fluorophore on the N-terminal and let it go in, if it is the full N-terminal, that is 1 to 40, the full-length peptide, you get a lifetime which is very, very short. While if you put it in the 10 to 40, which is the N-terminal truncated peptide, the lifetime is normal, like as if it's just in water. And the short lifetime is acquired only inside the cell. So that means something is interacting with the N-terminal as these things go inside the cell. Okay, that's where we are. And one of my students, Arpan, is breaking his back trying to find out what is it that this thing interacts with. But we do not know the answer yet. But whatever I have told you meets the first questions that we asked, which is whether this protein, which makes fibrils when there is a large aggregate, can also make small beta barrel-like structures. And the answer I have provided one by one is that yes, it doesn't prove it's a beta barrel because you cannot visually see it. In fact, we are working with one of my colleagues, Vinod Kumar in NCBS, to do some cryo-electron microscopy, try to see if we can see a proper barrel. But all the biophysical characterization that we have done shows that, yes, it's a very consistent picture that this thing is making a beta barrel in the membrane. Okay. Ajay, I'll ask you. I have a 5 minutes second part. Should I do it or should I not?
Yes, Professor Maity, please do it. Please do it.
Okay. So this is the new stuff that, again, one of my students who has completely gone orthogonal to the rest of the lab in doing something because he loves new things, Anirban Das, he has done. So now we are playing the Play-Doh with the protein. Okay. So imagine you have a small protein. This is a small protein called ubiquitin. Okay. It is ubiquitous in our cells. It does very important functions. And we looked at that. We asked this question that ubiquitin has these very well-ordered structures. And you can see one is there's one helix. There are a couple of other beta sheet-like things. When there are these arrows, they're beta sheet, and the helical is obvious. So we asked the question, suppose I take a part of ubiquitin, part of the protein. Again, a part of the protein doesn't fold, right? I told you because it has an inadequate number of residues. But I make it fold in the way it is folded in the original peptide or close to it by putting chemical constraints like staples on this peptide. So this is where this little part, this is again the N-terminal part, I put a staple, okay? Then I present it to the full protein. What do you think will happen? Is a part of the protein folded the same way, presented to the full protein. The first guess is nothing will happen because the protein is content in itself, right? So it's nicely folded and it doesn't care if something else which looks like a part of it is going around. Which is not completely true that proteins, we tend to think of it as very solid, nice objects, folded proteins, but they're actually frequently unfolding at least partially. But anyway, if it's reasonably stable, that probably nothing will happen. But suppose I jog it a little bit. I catch the protein while it is folding. So all proteins have to fold in our cells because they come out from the ribosome as a string, and it's like you drop that string in water and it tries to fold. So what happens if I put this pre-folded part in the protein, close to that protein, which is trying to fold, close to the ubiquitin? Well, what I didn't tell you is that, Anirban, as smart as he is, he has not just picked any part of it, he has picked the part which is known to fold first, and the rest of the protein folds around it. So the argument he made is that if I put this pre-folded part, which is called a folding nucleus, because the nucleus is folding, the rest of the protein might just grab it and fold around it, leaving as orphan its own nucleus part. So the protein has all the parts, including this part, but I'm supplying this. It's unfolded and I'm supplying this to it. And then it is possible that it will orphan its own part, the nucleus part, and take this pre-folded thing. Because protein folding is slow. The kinetic barrier is there where the first part, which is this N-terminal part, has to fold and the rest has to fold around it. But I've already presented it with a pre-folded thing. It's a decoy. It's something that is trying to fool it. So he tried doing this experiment and says, well, does it do anything? can I say if this protein is, this little decoy peptide is going and attaching to my protein when the protein is slightly destabilized? Okay, it's trying to partly fold and unfold. So we did some experiment with our colleague Ranavir Das, and he found out through his NMR experiments that yes, the colored region of ubiquitin, this is the whole ubiquitin, actually shows where the interaction of this peptide, which is the staple decoy, is with the rest of the protein, is exactly where the original folding nucleus sits, its own N-terminal sits. Okay, and when Anirban did his experiments, what happens when a protein is completely unfolded and you are doing the folding kinetics measurement, that is how quickly it can fold. If he presented this decoy peptide pre-folded, then it folded faster. It grabbed around it and folded faster. This is what is shown here. This is the rate constant for folding. This is the dose dependence of the decoy peptide. Externally, I'm adding this pre-folded thing, and then you see that the rate goes up by about 40% at about 60 micromolar. And it is not a non-specific effect. The decoy is recognized as its own part, because if you take this sequence and randomize it, then the effect, there's still a little bit of an effect, but the effect is much less. Okay, so this is a catalyst for protein folding. And imagine, this is a very interesting catalyst, because if you know how a protein folds and you know what is its initial folding nucleus, you should be able to then design a particular folding catalyst, a small peptide, for only that protein. Another protein which doesn't have that as a folding as a folding nucleus should not care about it. So there is a way to specifically design folding catalysts for individual proteins, even if these proteins are in a mixture. This is something that we are having a lot of fun with, variations of this, and hopefully this is opening up a new area for us, because this is just a peptide. So in principle, we can put these peptides also inside cells or express the peptides inside cell, thereby changing the kinetics of folding or the state of folding of a particular protein of our target. Okay, so that's it. I will finish and stop here. It's been 35 minutes. And let me introduce to you to the folks who are extremely talented. We're very, very lucky to have such talented grad students in our labs. Also, scientific officers. They're all extremely motivated. Barun did some of the Raman stuff that he talked about. Of course, there are past students like Devanjan who did the first parts. Shimli did all the single molecule stuff. And Bappa did that solid-state NMR, where we found out that it folds in an antiparallel way. Also did the fluorescence quenching by nanoparticles. And Anirban, I just talked about, has done this protein decoy kind of project. There are others who have helped. We usually have very small groups in TIFR, but they're all, they're one substitutes for 5. So very good, guys. I'm sorry. And of course, there are many collaborators without whom this wouldn't have been possible. And some of them are in TIFR, but they're all over the world. And our lab would not exist and would not be able to do anything if they didn't help. Okay, so financial support comes from DAE, Department of Atomic Energy, and the Department of Biotechnology. And thank you for sticking with me on a holiday. Thank you very much. I'm ready for questions.